Greetings. So with immune system, we talked about soldiers of the immune system, which is the immune cells, and we have the cytokines, or the chemicals that help us coordinate the attack. So let's talk about one of those cytokines, and it's called histamine. This cytokine is present throughout the body, but there's high levels or high concentration of this in three specific cells: the basophils, the eosinophils, and mast cells. And if you remember correctly, which cell line do they belong to? The myeloid cell line. And the Benz cells, the basophils, eosinophils, neutrophils. So those are the PMNs, right? So again, basophils and eosinophils, right? They have these granules, right? And then also basophils mature into mast cells, right? So what's the difference, right? So basophils, eosinophil, mast cells, they all have these granules. The difference is basophils and eosinophils have these lobulated, multi-shaped nuclei. The mast cells, on the other hand, do have the granules, but their nucleus is more defined. Okay, so again, mast cells, eosinophil, basophils are all granulocytes, and they have increased levels of histamine, and that's in the granules. And then, of course, basophils and eosinophils have that polymorpho nucleus, so they're PMNs versus mast cell, which is not a PMN. Again, you have more concentration of histamine in skin, lungs, and stomach. So what happens? Well, histamine gets released, right? And it acts on two receptors. It's an H1 receptor and an H2 receptor. And when it acts on these receptors, it does stuff. Okay, so we'll talk about that in a second. But the first question is, well, how does it get produced? How does it get released? So let's look at that next, and then um, we'll talk about what it does. So again, it's released, basically there's three ways it's released. The first one is type 1 hypersensitivity reaction. So you'd have an antigen, which is something which is external. And the adaptive immunity, remember adaptive immunity? And adaptive immunity to the external one is humoral immunity, right? And then humoral immunity is mediated by B cells and antibodies, right? So again, this is all a recap, and we'll talk about it in more details in subsequent videos. So again, an antigen gets processed by B cells, which creates an antibody, which targets this antigen. And this antibody now, what it does is it primes mast cells and sits on mast cells, ready and waiting. It's called sensitization. Okay. Now, when the antigen comes in again, right? Antigen comes in again, boop, it gets fit, it fits in very, very nicely here. Um, Fits in very nicely on these antibodies and degranulates the mast cells. Degranulates the mast cells, and now you have lots of histamine around. Okay? There's tissue injury. Your mast cells degranulate. You have your mast cell, okay? And you have this bad guy, the bacteria, okay? And it produces endotoxins which lyse it, and it releases histamine. Last but not the least, you also have drugs and toxins. So now I have histamine that got released. It acts on two receptors, H1 receptor and H2 receptor. There's other receptors as well, but these two are what I call need to know. So what does H1 receptor do? Okay, it causes capillaries to dilate or blood vessels to dilate. Okay, so what does that mean? So this is my blood vessel. Okay, and it's a pipe. It's got a fixed diameter. It actually dilates it. The diameter increases. Well, what happens when the diameter increases? You will have more blood flow. Okay, have more blood flow. More blood flow means more heat and more redness. Okay, so capillary dilation leads to more blood flow, leads to more heat and more redness. Okay, it also makes the capillary permeable. So usually the endothelial is closer together, but what this does is this increases the gap between the two cells. So that allows more fluid to come out, more stuff to come out. It's like a pipe. So first you increase the diameter of the pipe, and you're going to have more fluid that can go through the pipe. Now we create some big holes in the pipe, so there's going to be more fluid that's going to come out. So again, it increases the permeability. So what does that mean? That means more fluid is going to go from inside the blood vessel to outside the blood vessel. Okay? Or goes more from intravascular in the blood vessel to interstitial, which is outside the blood vessel but also outside the cell. Okay? Now, there's going to be some pain, so it does mediate with pain. And more specifically in lungs, it causes smooth muscle constriction. Okay? Now, this sounds very similar, right? Sounds very similar. So the concept goes back to inflammation, right? Which is a big, big way of how the immune system works. Inflammation is what? Essentially, at the core of it, it's an immune response designed to clean debris. And once you clean the debris, it's to help the body regenerate. So, whenever there's inflammation, the histamine is released, it acts on H1, it causes capillaries to dilate and increases heat in that area and redness in that area. It causes edema because there's more permeability. There's pain and itchiness, okay, which is on the skin. So these are some of the signs and symptoms of inflammation that one gets to see, okay. What else does this histamine do in the immune response other than the inflammatory response? It attracts neutrophils and macrophages. So remember, neutrophils and macrophages. Both are myeloid lines, right? Myeloid cells. Neutrophil is a granulocyte. Macrophage is not a granulocyte. Okay? All right. So how does it do that? So let's take a peek at it. It's a very interesting way it works. Okay? Let's just see how it works. Okay. So this is your blood vessel wall. Okay? See? This is the blood vessel wall. Okay? And this is all in the blood vessel. So now that the diameter has been widened, there's more blood that's flowing at a higher speed, right? Lots of flow. And then you have macrophages and neutrophils and a lot of other WBCs hanging in there, okay? And they're going at pretty fast rate. So what happens? Well, if you go very fast through the flow, you can't get, it's difficult for you to get on the edge and get to the site where the histamine is getting released. The goal is to get these cells to the site of histamine release because that's where the need is, right? So how does one do that? Well, histamine and other mediators act on these cells in the endothelium, okay? And essentially they raise these proteins, and it's the best way to kind of create a shape. It helps me visualize things better and explain it better. And then these white blood cells also have these hooks in there. Okay. So essentially, this white blood cell is going very, very quickly. There's a lot of blood flow, but then these these mechanisms, what they do is they hit this. So what happens when they do? So this is an example. So these two are logged, and this rotates. Okay. And so what are the two things that that does? A, it slows it down, causes it to roll, and move it closer to the wall of the vessel. Okay, and this happens a few times, and it rolls and it rolls and it rolls, and it comes closer to the vessel. So the margination is the process where WBCs actually get closer to the wall, and there's rolling that happens. So there's margination and rolling, two things that happen. Okay. Now, once that we are able to get this white blood cell close to the wall, even though there's flow, you can see that this attachment prevents it from just flowing away. In fact, it helps it rotate. Now, the other thing that the histamine, cytokine, and other cytokines do is they activate the same kinds of protein on these gaps. Okay? So remember, so we have this that is rolling around when it's getting close to the cell wall, when it gets close to the endothelial vessel wall, right? And then this rolls into this gap. When it rolls into the gap, again, it gets stuck in this protein, okay? So what does this prevent this, what does this attachment on the side in the gap prevent it from happening? Again, if there isn't an attachment, this can flow through as it's rolling, but this attachment prevents it from doing that. So again, it rotates, gets in here, doesn't go with the flow, and squeezes out, and this is called migration. So it actually rolls. So again, it's going fast. It hits these attachments, it hits these proteins, and it does it a bunch of times. It gets slowed down, it rolls, it comes close to the vessel wall till finally it's close enough and slow enough to where it just gets stuck, rotates, goes in through the gap, and then gets stuck in the gap and then migrate. And this is actually how um, histamine promotes macrophages and neutrophils from migrating to where they need to go. Okay? So that's what histamine H1 receptor does. Now, there's an H2 receptor as well, and this is in the stomach, the parietal, this is again in the stomach. So this is the stomach lumen, okay? So this is where the food comes from, the esophagus, okay? And so what happens is, again, there's a mechanism by which there's cells in the stomach that get stimulated that produce histamine, and that histamine then stimulates the H2 receptor. What the H2 receptor does is it promotes secretion of H plus or hydrogen ion into the stomach lumen. And there's an exchange where the H plus or hydrogen ion, which is actually a proton, because hydrogen is one electron and one proton. So once you take the electron away, it becomes positively charged because all you have is a proton. So again, you have the hydrogen ion that gets into the stomach lumen, and the potassium ion, which is a positive ion, goes back into the stomach. So again, it's an exchange of charges, so to speak, right? And the proton gets into the stomach, and the proton combines with hydrochloric acid, with chloride ion to give you hydrochloric acid. So again, proton pump is what this mechanism is, and we'll study more about it in GI. But essentially, that is the purpose of the histamine there, is to stimulate the H2 receptor, which promotes acid production in the stomach. So let's go ahead and recap real quick. So again, what did we talk about in this video? So we started to talk about A, what is histamine, and where is it most located? Okay. Then we talked about, well, how's it released? We talked about once it's released, which receptors does it act on? And then we talked about two receptors. We talked about H1 receptor, where it causes cardinal signs of inflammation: capillary dilation, capillary permeability, pain, and lung smooth muscle constriction, which causes the heat, the redness, the edema, all of those signs of inflammation. And we also talked about how it attracts white blood cells with certain processes like margination, rolling, um, you know, and then we talked about adhesion, diapedesis, which is the WBC actually squeezing through the gap. So again, we talked a little bit about that, and and then finally we finished up with the H2 receptor, which is the other receptor, and when the histamine molecule acts on it, then what does the parietal cell, what does that cell do, and how it causes the acid production.